Hello, everybody. My name is Pranati, and I'm a final year dual degree student in the Department of Complex Systems and Dynamics. I have worked on my dual degree project under the guidance of Professor Karthik Raman at the Computational Systems Biology Lab in the Department of Data Science and AI at the Indian Institute of Technology, Madras. For my dual degree project, I have worked on the comparative analysis of microbial community structure across human and environmental microbiomes. So, what is a microbiome? A microbiome is a community of microorganisms that exists in a particular environment. These microbiomes have been found in diverse environmental and host associated conditions on the planet. They have been found in the ocean, fish gut, and human oral microbial communities. And in this project, I am trying to understand the interaction patterns between different bacterial species and how they vary between host associated and environment associated samples. So, the overarching goal of this study is to understand if microbial communities from very different environments follow universal organizing principles or if each community is entirely unique, and to understand if the taxonomic level at which this is studied affects the results directly. So, what was the overall approach used to study this? We've used the Microbe Atlas dataset, which is a publicly available harmonized dataset where for each samples we know the abundance of bacteria present, and this has been studied at both the genus and family level after removing rare taxa to remove noise, and statistical and community level analysis was performed, followed by network analysis to get a holistic view of different community structures. So, what did we find out from our results? We saw that microbial communities clustered differently depending on the environment, with human associated microbiomes forming tighter and more stable groups. We then explored whether microbial communities assemble randomly or through ecological selection, and we saw environmental microbiome showed greater variability and weaker clustering, suggesting that different environments shape microbial community assembly in distinct ways. We constructed microbial co-occurrence networks to study how taxa interact within each ecosystem, and we saw that all microbiomes showed modular, non-random organization shaped by ecological interactions. We then constructed directed microbial networks which revealed interaction patterns shared across ecosystems. Reciprocal interactions were consistently overrepresented across microbial communities. Finally, we also explored whether microbial communities exhibit higher order interactions beyond simple pairwise relationships, and these higher order interactions varied across microbiomes and revealed ecological relationships not captured by standard networks. Together, this suggests microbial communities are organized through complex collective interactions. This work shows that microbial communities are shaped by both ecological context and host association. And environmental microbiomes exhibit great heterogeneity and more complex interaction structures, while all microbiomes show organized, non-random network architectures linked to stability. Finally, incorporating directional and higher order interaction provided additional ecological insights beyond conventional pairwise analyses and helped move towards a more system level understanding of microbial community organization. Thank you.